So before we take a short break, I will talk about the homogeneity of alginate gels, because depending on how you make them, they have different type of homogeneity. The reason for this is that the formation of gels and beads involves diffusion. So if here you have a alginate drop that just dropped into calcium, the calcium must diffuse into the system, and that takes time. And at the same time, the alginate, as it reacts with the calcium, you form a chemical potential gradient. So alginate will diffuse towards the gelling zone. And therefore, you have a high concentration of alginate at the surface, and in the middle you have the lowest, because it migrates from average value to higher value and lower. This gradient is sometimes something you want, in other cases sometimes things you don't want. It depends what you have inside the bead. And there is a lot of discussion about human cells, what kind of mechanical properties they want to have. So being able to tailor the elasticity inside the gel can be important. So here is one example how we can use advanced technique, in vitro epimerization, to control homogeneity. And we have used, in this case, fluorescent labeled alginate, which we can observe by confocal microscopy. I got this from my colleague, Professor Gudmund Skjogbræk. This is an alginate bead where we have, by some tricks, made it quite homogeneous. Actually, too homogeneous for certain purpose. And here you can see the fluorescence intensity across the profile is fairly constant. A little dip in the middle. Compared to an alginate which has been epimerized a little bit, you can clearly see the gradient, and this is how it looks by measurement. So this is one way to manipulate the alginate and the homogeneity. To start, so this second part of the lecture, I will finish off with the alginate, and I will then switch a little bit to chitosan. That's what we have time for today. First of all, calcium alginate gels are unstable under physiological conditions. If you make an alginate bead and try to put it inside the body, it will not be stable. The reason is that physiological conditions means 0.9% sodium chloride, 15 millimolar sodium. And sodium will compete with calcium because of the concentration is so much higher. So we have a calcium-sodium exchange. So we lose calcium, and therefore the gel bead will swell and dissolve, although slowly. In some cases, we need to use covalent cross-linking. Then we have to add something binding to alginate. This is possible in some cases, again depending on the system. As we saw just before the break, in vitro epimerized alginates can can influence homogeneity, but it can also limit swelling. And I include a quite recent study from Trondheim, where we took first a standard high G alginate, 69% G, and made beads from that. And we transferred that to fresh sodium chloride up to 8 times and measured the diameter. And you can see that every time you move the bead to fresh sodium chloride, it gets a little bigger. So it swells. It loses calcium, attracts water, and if we will go for a very long time, the capsule will dissolve and go away. But if we made an alginate, which was epimerized first with Alg4 and then Alg6, 2 enzymes, so we have about 54% G in the end, we have an alginate which is very, very stable, like this. And this is now used in some quite advanced studies I will show later. Another way to make completely homogeneous alginate beads, if you want that, is either to use sodium chloride or mannitol, some osmolite. I will not explain why, but this works. You can make homogeneous. Or you can use a very common method, the GDL, calcium carbonate method. This is much used in many laboratories because you make a nice and homogeneous gel. I will try to explain. It is often called the internal calcium release. So in the previous case, the calcium came from the outside and diffused into the gel bead. In this case, we will try to have calcium in place before gelation. And the trick is to use calcium carbonate and this compound GDL. GDL is an abbreviation for glucono-delta-lactone. And a lactone is the same as an internal ester. And an ester is a reaction between carboxylic acid and an alcohol. And in this case, it's D-gluconic acid, which is very similar to glucose, except that this is carboxylic acid instead of aldehyde. So this can make an ester like this. And this compound is actually used in the food industry to manipulate pH, because when you dissolve this material, it is neutral, but it will slowly hydrolyze in water, forming carboxylic acid, and forming carboxylic acid leads to a decrease in pH. So what do? The alginate solution is mixed with calcium carbonate in the form of finely ground powder. You know that calcium carbonate is insoluble in water, I hope. It's the same as limestone. But by adding GDL, the following happens.